Hi, we're team number 2, and today we're going to present our PPT of cloning. Okay, so we can describe cloning as a process of generating genetically identical cells of an organism in nature. There are some species that clone themselves as reproductions, and there's known as sexual reproduction, for example, bacteria. Although prokaryotic cells have the ability to recopy themselves, eukaryotic do not. Eukaryotic cells reproduce themselves through cell division carried out in a process called mitosis. Cloning is a natural process in some cells, but not in everyone. That's why it's a discussion topic. Nowadays, scientists use cloning to replicate the DNA of an organism. Having the bioethical issue with it is moral to replicate DNA of living things that are not natural created to be cloned.
Types of cloning. Well, there are 3 types of cloning. The first one is gene cloning, the second one is reproductive cloning, and the third one is therapeutic cloning. Gene cloning. Well, it is a process where a specific gene is copied with the purpose of forming new organisms with the same characteristic. There are 5 steps. The first one is that the DNA is extracted from an organism to take a specific gene and cut it into gene-sized pieces with restriction enzymes. The second one is that bacterial plasmids are cut with the same restriction enzyme. The third one is that gene-sized DNA and cut plasmids are combined into one test tube. Repeatedly, a plasmid and gene-sized piece of DNA will anneal together, forming a recombinant DNA. The fourth one is that the recombinant plasmids are transferred into the bacteria with the use of electroporation or a heat shock. The fifth one and the last one is that the bacteria is played out and allowed to grow into colonies, and they are called gene library. It's important to mention that the gene library identifies the colony containing the gene of interest by looking for one of 3 things. The first one is the DNA sequence of the gene of interest or a similar one, or a protein encoded by the gene of interest, or a DNA marker whose location has been mapped close to the gene of interest.
Now I'm going to talk about the reproductive cloning. And reproductive cloning involves the implantation of a clone embryo into a real one or an artificial uterus. The embryo develops into a fetus that is then carried to the term. Reproductive cloning experiments were performed for more than 40 years through the process of embryo splitting, in which a single early stage 2-cell embryo is manually divided into 2 individual cells and then grows as 2 identical embryos. And we know that following the birth of Dolly, who was generated through the process of SCNC, which is a process that entails the removal of the entire nucleus from a somatic body cell of an organism, followed by insertion of the nucleus into an egg cell that has had its own nucleus removed. And once the somatic nucleus is inside the egg is stimulated with a mild electrical current and begins dividing. Thus, a clone embryo, essentially an embryo of an identical twin of the original organism, is created.
Now we have Therapeutic cloning. This is the creation of a clone embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. The applications are these stem cells can be used in experiments aimed at understanding diseases and developing new treatments for them. Two types of adult stem cells are like the stem cells in the blood forming bone marrow and skin stem cells are the only two stem cell therapies currently in use. And the procedure is very simple, could be like to explain. It's basically called nuclear transplantation to produce a stem cell, in where the diploid nucleus of a human somatic cell is introduced into an enucleated ovule that will be stimulated to initiate cell division. The embryo will never be implanted in a foster mother. This is how it differs from reproductive cloning. I'm going to show you this diagram. Here you can see that first we have our somatic body cell with the desired genes, and we also have the ovule. And then we're going to get only the diploid nucleus of the somatic body cell. And we're going to get out in the other side the nucleus of the ovule. Then these are going to be joined. And this is the somatic cell produced, like in here you need to stimulate it to initiate the cell division. Then you have your clone, and that can be used to all the experiments, like doing tissue culture, for example.
Now, a real-life example of the cloning techniques applied is the case of Dolly the sheep. In 1996, a group of researchers from the Roslin Institute in Edinburgh, Scotland, first applied a somatic cell nuclear transfer technique in a sheep called Dolly. To do this, they used two sheeps, one with a white head and one with a black head. From the white-headed sheep, they took a somatic cell from the mammary gland and removed its diploid nucleus. On the other hand, from the black-headed one, they took an ovule and removed its haploid nucleus, resulting in an ovule without nucleus. Subsequently, they inserted the diploid nucleus into the enucleated ovule to form a zygote, to which they then applied an electric shock. Can you please go to the next slide? They applied an electric shock to stimulate the cell division process, and the resulting embryo was cultured in vitro until it reached a stage where it could be transferred to a third sheep that played the role of a foster mother. The embryo developed properly, and the resulting sheep was identical to the donor of the nucleus, which was the white-headed one. The next slide. This technique has allowed the obtaining of clones of other animals, such as mice, pigs, horses, dogs, and cats. However, the percentage of success in cloning is low, approximately 2 or 3%. Cloned individuals have a high probability of having genetic defects, and their health is diminished compared to animals born from sexual reproduction. In the case of Dolly, she had health problems since she had a really short age, because she exhibited arthritis and when she was 5, and at 6 she had to be sacrificed because of lung cancer that she developed because of an infection. So this is really bad, because the average life of a sheep is from 11 to 16 years old.
The ethical controversy. The ethical controversy over the ethics and therapy of research cloning. Some individuals have an objection because it is considered that the manufacture and the destruction of the human life, even thought that the life has not developed. But in other hand are those
who support the therapy cloning believe that there is more imperative to heal the sick and to seek rather the scientific knowledge. However, the opinion of each individual, they are laws, an international convention that attempt to uphold certain ethical principles and the regulation concerning cloning.